The last section of what we need to do in genetics is talk about mutations, then a little bit about artificial selection and genetic engineering, and then we'll be done. So, we're going to go over a question today. What is a mutation?
Genetic abnormality.
That's a change in the gene. So what does that mean?
It's all like
What actually happens when a mutation has taken place?
the mass is different now.
We know about the biochemistry of DNA now. So what's actually happening? Grace?
Isn't it that the DNA copy is wrong, and that the change is kind of, it keeps the copy the same way, so that's
Yeah, a change in what? Carmen?
Order of
Yeah, change in the order of what?
the mRNA. The
night phosphate.
that's
thinking of DNA.
sometimes it could be like
What's changes? So we need to flash back here to biochemistry. A DNA molecule is made of what? What is the monomer of a DNA molecule? A nucleic acid, a nucleotide. DNA is a nucleic acid. Nucleic acids are made of nucleotides. Nucleotides have 3 parts to them. They have the sun,
Phosphate group
the house,
and the garage.
but what are they? Phosphate,
Oh,
sugar,
no.
and a base. What are the 4 DNA bases? Okay.
And that last
A,
one.
T, G, and C. A DNA molecule is made of
And that last one.
hundreds of millions of these nucleotides strung together. The pattern, the A, T, G, C, those are the pattern that they come in. Every 3 is a code for one what?
G.
Nope. 3 bases is called a codon. It is a code for
A protein.
no,
Amino acid.
1 amino acid that's going to go into the protein. Every 3 bases is a code for one of the 20 amino acids. ATG is a code for 1 amino acid. CCC is a code for a different one. As those bases are read, they're used to assemble a protein made of many thousands of amino acids strung together. All right, so a mutation is when in the copying of the DNA molecule, which normally, under ideal conditions, gets copied exactly. It replicates. You have one DNA molecule, one side of the chromosome produces the other half identically. But sometimes mistakes happen. That's a mutation. Sometimes a G gets substituted for an A. Sometimes a new base gets inserted in. Sometimes the base is just taken out. There's different types of mutations. So a mutation is a change in the nucleotides that make up the DNA of an organism. Mutations are,
Wait, Mr. Buros, are you going to have review classes for like the SAT and stuff?
no, we'll review in class.
Is
Um,
that what kind of review class after school?
I don't believe so.
And
So about, a mutation happens about 1 in 100 million bases. So when bases are copied, about once every 100 million bases, something happens. A mutation takes place. So they're relatively rare. Okay? And obviously by changing one of these bases, that could change what? If I change a G to a C, whoop, yeah, that could be a code for a completely different amino acid. And then you have a different amino acid gets put into the protein, and then the protein is obviously different. Okay? And so that can alter how that protein works. proteins are the structures of our cells, they are enzymes, and so if the shape of the protein is incorrect, it may not work properly. And so that can lead to problems, okay? It can lead to a protein not functioning properly and harmful traits. Also, once in a while, it can lead to a beneficial trait. So sometimes this new protein, maybe it works better. Maybe it codes for some darker fur color now, and that conveys an advantage to the offspring. So there are times when a mutation can result in a beneficial trait. Sometimes it could result in a harmful trait. Sometimes it doesn't change anything at all. Because remember, there's several codons for the same amino acid. So sometimes a codon can get changed, a nucleotide change, and it still makes the same protein. The DNA itself, most of the bases in our DNA are non-coding sections of DNA. About 80% of the letters are not actually part of the code for different proteins. They're non-coding DNA. So if the mutation happens there, it may not have a significant effect on a protein. Now, if a mutation happens in a skin cell, say that your cells, or the cells of a person are reproducing. You produce new skin cells constantly. Let's say in that process, a mutation takes place in a skin cell. Okay? That's a somatic mutation. It's a mutation in body cell. It can be damaging. For example, exposure to ultraviolet radiation increases the rate of mutations. It can lead to cells starting to grow uncontrollably. If that happens in a person's skin, it can lead to skin cancer. Can that be passed on to offspring? Can a mutation in a skin cell be passed on to the offspring? Because the only thing passed on from parents to offspring are the sex cells, are the actual DNA and chromosomes in a single sperm cell and a single egg cell. If there happens to be a mutation in the production of a sperm or egg cell, that's called a germ cell mutation. And that is something that can be passed on to offspring because they inherit those chromosomes from their parents. So there's two sort of groups of mutations. Alterations to the chromosome itself and gene alterations. So chromosomal alterations are a change in the structure of the actual chromosomes. And obviously, when a whole chunk of a chromosome gets altered, that affects many, many genes, because the chromosome carries many genes. And so, causes significant effects. Some examples. Non-disjunction. We know that during meiosis, producing sperm and egg cells, chromosomes line with their homologous partners in tetrads. Homologous partners get split up, one chromosome going to each cell, and that's what reduces the number of chromosomes in half. Well, once in a while, the two homologous chromosomes do not separate and each go into a new cell. Sometimes non-disjunction takes place, that the chromosomes don't separate and they both go to one cell. And what that leads to is an extra chromosome in that sperm or egg cell. Okay. And the opposite cell that didn't get it has one less chromosome. That's what non-disjunction is. And that can lead to some syndromes that we're familiar with. Down syndrome, for example. Down syndrome is when you say the word syndrome, it means a whole bunch of different effects create what we call a syndrome. So Down syndrome is caused when a person has an extra 21st chromosome. So sometimes the 21st chromosome has non-disjunction. Two of them go to a sperm cell or an egg cell. If that sperm or egg cell produces a zygote and it grows into an embryo and a fetus, then that child would have Down syndrome. And having this extra chromosome produces extra proteins in all of the cells of that person, and it leads to a whole host of effects that we typically think of as Down syndrome. Physical effects, delays in development. Okay. So there's that likelihood of non-disjunction taking place does change with the age of especially the mother, but also with the father. As mother gets older, if you look at a graph showing the incidence of Down syndrome with maternal age, which you'll see as it gradually goes up. So the older a woman is when she becomes pregnant, the higher the chances of having a child with Down syndrome. There's a more likelihood that that those eggs, when they mature, non-disjunction happens and there's an extra chromosome. There's other chromosome abnormalities as well. Klinefelter syndrome is when a person is born with two X and a Y chromosome. So an extra chromosome, an extra X chromosome. A person with Klinefelter syndrome is a male that has a syndrome, meaning a whole host of effects that come with having an extra X chromosome in a male. And it results in usually infertility and a whole host of other things. Turner syndrome is another chromosome disorder, and that's when a woman is born but with just a single X chromosome. And again, leads to a whole host of different things that are accounted for by that X chromosome. So a male with Klinefelter syndrome, having an extra X chromosome, leads to lower productions of testosterone. A female with Turner syndrome has lower production of estrogen. So those are some examples of non-disjunction. Now, you don't end up with, in humans, people being born with 3 or 4 or 5 extra chromosomes, or missing 3 chromosomes. Because when you are missing that many chromosomes, the resulting zygote is not viable. It never grows, it never starts to reproduce, because there's too many chromosomes, or too few chromosomes, and it just doesn't survive. So usually it's plus or minus 1 chromosome is about the limit, and then the offspring never grows. So there is though in plants, polyploidy is somewhat common. So we know what ploid means, like haploid, diploid. What do you think polyploid is? Yes, several copies of chromosomes. Sometimes plants can be 3N, triploid, or 4N. In fact, so higher animal is never born with 3 or 4 sets of chromosomes. They're not viable, but plants are. In fact, often this is induced in plants, because plants that have polyploidy are often larger, grow better. Seedless watermelons, for example, oranges, are often produced from polyploid plants. They produce larger fruits. So it can be induced using certain chemicals. And then translocation is when a part of a chromosome swaps to another. This is translocation, where you have your normal chromosomes, but sometimes, so normally we would call that what, if it's 2 homologous chromosomes? Crossing over. But sometimes that same process happens between non-homologous chromosomes, and that's what translocation is. And that leads to problems in the chromosome. Here we have again the 3 21st chromosomes that results in Down syndrome. And here are some of the effects of that.
Say that you had a mutation that produced so much extra iron in your body that you had iron claws coming out of your head. Would that be possible?